bound receptor, known as a metabotropic G protein receptor, is found in the cell membrane. The top part of the receptor is known as the receptor site. This is where epinephrine, known as a ligand, attaches to the receptor and begins a process called protein conformation. This is when a protein changes shape. When the ligand attaches to the receptor, it activates the G protein. The G protein is heterotrimeric, meaning that the protein is broken into 3 subunits. These are the gamma, the beta, and the alpha subunits. The G protein disengages from the receptor. Guanosine diphosphate is then exchanged for guanosine triphosphate, which activates the alpha subunit. The alpha subunit separates from the other subunits and attaches to the adenylyl cyclic. Activates the enzyme, which uses ATP or energy to make cyclic AMP. Cyclic AMP is the second messenger in the signal transduction process. AMP then go to the protein kinase A, where it binds to the regulatory portion of the protein kinase. When this happens, it releases the catalytic portion of the protein kinase. ATP then activates the catalytic portion in a process known as phosphorylation. They can then act on molecules in a cell, such as phosphorylase, which results in a number of cellular responses. Phosphorylase can then react with other molecules in the cell, such as glycogen. When phosphorylase reacts with glycogen, it sends glycogen out of the cell. Glycogen is disengaged from the G protein receptor. Phosphodiesterase is released to get rid of any remaining cyclic AMP. This then causes the catalytic part of the protein kinase A to reattach to the regulatory port of the kinase. ATP of the alpha subunit then loses a phosphate, returning it back to GDP. The alpha subunit disengages from the enzyme. This deactivates the enzyme and stops production of cAMP. The alpha subunit then reattaches to the other subunits of the G protein. These then reattach themselves to the G protein receptor and are ready to begin again.